This is part 2 of cell communication. In this lecture, we're going to cover enzyme-linked receptors, and we're going to use what's known as the EGFR pathway as our example of a receptor tyrosine kinase cascade. So let's talk about enzyme-linked receptors. Enzyme-linked receptors are proteins. We have one up here, right here, the top picture here. It's a protein that spans all the way across the phospholipid bilayer of our cell membrane. It has an intracellular domain, that's this pinkish region down here, and an extracellular binding domain. So this is what's known as an integral protein. It spans all the way across the membrane. Sorry about that. It expands all the way across the membrane and will change shape when activated. So the extracellular region or the extracellular domain of this protein is where the signal or ligand will bind to the receptor. And the intracellular domain is known as the catalytic domain. This is what will actually catalyze or begin our intracellular or signaling pathway. When a enzyme-linked receptor is bound by its signal, it activates this intracellular catalytic domain. So here in the bottom left, our receptor is inactivated. You can see our ligand is on its way in to bind to it. Near our enzyme-linked receptor, near the intracellular catalytic domain, is some type of protein. And when our signal binds to our enzyme-linked receptor, the receptor will change shape. That change in shape will cause the transformation of ATP. Now, ATP is a signaling molecule. This is one of our big energy molecules that we use in the cell. And so ATP, a phosphate group, one of these little yellow balls right here, a phosphate group will be broken off of ATP to make a molecule called ADP. ATP stands for adenosine triphosphate. ADP is adenosine diphosphate. When this phosphate is broken off, it is given to or added to this purple protein down here. When it did not have a phosphate, it was known as unphosphorylated. Here, a phosphate is added to it, so it's referred to as phosphorylated. ADP is going to go somewhere else in the cell to go find another phosphate. And this now phosphorylated protein is now activated. And this phosphate right here is going to get passed like a hot potato from one enzyme to the next in order to activate each of these enzymes to carry out the cell signaling pathway, which remember is part 2 of cell signaling. Now, these types of proteins here that receive and donate these phosphates are referred to as kinases. They are a class of proteins that transfer phosphate groups from very high energy molecules to lower energy molecules. Nowadays, sometimes they're called phosphotransferases to keep with the naming, the way of naming proteins and enzymes. But in the old days, they were called kinases, and they are still referred to that. So a kinase is a protein that's going to take a phosphate group from a high energy molecule like ATP or GTP, and it's going to rip off that phosphate and transfer it to some other protein of lower energy. Now, when that is done, when that transfer is done, remember, some of that energy is going to be used to do some kind of work. So here are receptor tyrosine kinases. Their primary function is in hormone signaling. They are found in all animals and choanoflagellates, but they are not found in non-animal eukaryotic cells like plants and such, and they are also not found in prokaryotes. The receptors are not activated until they are dimerized. A dimer is simply when two proteins attach to each other. So down here, let's take a look at this diagram. On the left, all the way to the left, I have two unbound, my mistake. To the left, I have two unbound receptor tyrosine proteins. Okay, I have two receptors here. I have two ligands that are going to bind to these two receptors. When both ligands are bound to both receptors, these two receptors will come together. When they come together, this is known as dimerizing. Now, if they are a homodimer, it will be two of the same receptors bound to each other. If it is a heterodimer, then it will be two different receptors bound to each other. Once these two receptors are bound to each other, these are receptor tyrosine kinases. So since they are kinases, they are going to grab a bunch of phosphate groups off of ATP. In order for the receptor tyrosine kinases we are looking at to be completely activated, they have to actually take 6 phosphate groups. So it requires 6 ATP molecules to activate these receptor proteins. Once these receptor proteins are activated, relay proteins, other kinases, are going to attach to all of these different phosphate groups and start carrying out the signaling cascade or the signaling pathway. Now, once activated, that intracellular domain recruits all those other proteins. Right? Go back here. All of these proteins got recruited because of these phosphates on there. So when they get recruited, these are referred to as relay proteins. They are basically, think of a relay race when somebody passes one baton to the next runner and he passes it to the next runner and she passes it to the next runner. So this constant passing of the baton is a relay race. So a relay protein is a protein that's simply going to take a phosphate from one area and transfer it to another. The first protein in this relay race, the first relay protein, activates or begins that signal transduction pathway. A whole series of proteins, what's known as a cascade of proteins, is going to follow the primary initial relay protein. The transduction pathway that is carried out and activated by this relay protein is known as a protein kinase cascade. Allow the name of it to tell you what it's doing. So it's made up of a bunch of proteins. The class of proteins are kinases. When you hear protein kinase, you know that proteins are moving phosphate groups around automatically. When you see kinase, you know that a phosphate is involved. You know it's a cascade, so it's going to be one protein must follow the next, must follow the next, must follow the next. It is in a very specific order. The example we're going to look at is the epidermal growth factor. EGF is a hormone protein, and it's a ligand that activates an RTK pathway. This RTK pathway will cause cells to enter into what's known as proliferation and differentiation. Proliferation just means excessive cell growth. So the cell it'll kind of hijack the cell into the cell cycle and increase the mitotic rate of the cell, increase the cell's division so the cell becomes more cells. Differentiation means that it causes a change in the function of the cell and causes it to start doing different things. Now, the one we're looking at, EGF, the reason we look at this pathway is because it is highly active in different forms of cancer. It is a growth hormone. And in some cells, the growth hormone should be kind of turned off. The receptors for that are tied off or turned off because we don't want those cells rapidly progressing. But some cells, if they have mutations, their receptors will either be activated or they'll be of the receiving mode or capable of receiving EGF. Other cells that may be mutated may be overproducing epidermal growth factor. So when that epidermal growth factor gets produced in really high concentrations, the cell itself will be over-signaled or overstimulated. The overstimulation causes the cell to divide very, very rapidly. And the more quickly the cell divides, the more mutations it will begin to accumulate, and the cell will then become cancerous. As these cells divide rapidly, they start kind of clumping up together, and we start seeing tumor formation. So that's why we're looking at the EGF pathway. So the EGF receptor, this is a little tricky to see here, but here in the top left corner here is our EGF receptor. And of course, here is our epidermal growth factor itself. So this blue area here is referred to as the EGFR. So the epidermal growth factor receptor, and then the red dot up top here is EGF. When EGF binds to its receptor, we have dimerization. So the two receptors come together. When these two receptors are dimerized, they start ripping off phosphate groups, so we have some phosphates here. And our first relay protein in this here is called GRR2. GRR2 will recruit a second protein called SOS. So we have down here in the left in written form the series of proteins. So GRB, which is GRR2 in here, that's the same protein, GRB will recruit and phosphorylate our SOS. So it starts out as GRR2, gets a phosphate group, becomes GRB. GRB will bind the SOS protein, and SOS in turn will bind a protein called RAS. RAS has a GDP molecule, and the phosphate that it gets from these relay proteins will, oh my goodness, I'm so sorry. The phosphate it gets from these proteins over here will turn the GDP bound to it to GTP. Once RAS has been activated, it activates RAF. It hits the kinase cascade. So this phosphate group starts to get tossed around. RAS activates RAF to MEK to MAP kinase. MAP kinase enters into the nuclease and is able to activate transcription factors for cell division and cell differentiation. Over here on the left, this is just the list of actions that occur in this kinase cascade. One thing I want you to notice here is what's called the RAS-RAF-MEK. The RAS-RAF-MEK pathway over here. The RAS-RAF-MEK pathway. This particular pathway is used by multiple cells, lots of different cells in lots of different signal transduction pathways. They are common proteins, and they are always activated by this whole GRB SOS process, which are activated by RTKs. Not every RTK is an epidermal growth factor receptor. Okay, I want you to understand that these proteins, these signaling proteins, are used in multiple different pathways, but they all use the same type of mechanism. So they all use this same RAS-RAF-MEK pathway. Just what the MEK activates in a liver cell is going to be different than the transcription factor that's activated by that pathway in a kidney cell. So they use the same mechanism, but they activate different things in those cells. You should be very familiar with this pathway and how they work here, because you may be asked questions about, well, what happens if we mutate the MEK protein? Or what happens if SOS doesn't have a phosphate to give RAS? What's going to happen to the cell? You need to look downstream in these pathways. Those are the types of questions you'll see on an exam and that you should be thinking about. Thinking about what happens if I interrupt one of these steps in this pathway. What happens downstream from the pathway, and what might happen upstream. What happens if these molecules here, these RTKs are constantly activated, but SOS is not functioning properly? What could possibly happen to the cell? Okay, not just to the receptor. Here's just another diagram from a cancer article that I had read that shows this RTK pathway. So this is an RTK pathway, and it's we've got some other type of growth factor. This is not epidermal growth factor. It's a different growth factor, but it's the same idea. It's another hormone signal. Here's our two receptors, right? Our two RTKs, and these have this phosphorylation process here. We have more relay proteins, and here's our GRB2. So we are using the exact same, right, RAS-RAF-MEK pathway. We're using this same signaling pathway. However, it's being done through a different growth factor pathway. So it's a different hormone. Okay. Over here on the right is a G protein receptor, and we're going to talk about those in the next lecture. I like this particular diagram because it's showing you both the RTK pathway and what's called a GPCR pathway. And this pathway here oftentimes can interrupt or enhance an RTK pathway, and that's what's happening in this particular type of cancer. So we have, again, our growth factor ligand, our hormonal ligand binding to two RTK receptors. They dimerize, come together, and become phosphorylated. A set of relay proteins bind to these receptors and recruit our first relay protein in the pathway GRB2. This activates SOS and RAS and RAF and MEK, and we move into transcription factors. This is a copy of the image from your textbook of the EGFR pathway. So here's our EGF hormone and here's our receptor. Here are the 6 phosphate groups that bind to EGF receptors. When 6 phosphates are bound, GRB2 will finally bind, activate SOS, and here's RAS. Now RAS has what's known as a guanosine diphosphate. This is similar to ATP, it's just called a GDP. And this RAS protein has this GDP attached to it. The phosphate group that GRB transfers to SOS is then transferred to RAS and changes the GDP to a GTP. The GTP is going to be then passed off to the RAF1 protein, and this RAS-RAF protein will now recruit a MEK and become the RAS-RAF-MEK cascade. This activates these MAP kinases, and the MAP kinases move into the nucleus and they start to activate these different transcription factors that are found on the DNA in the nucleus. These transcription factors will turn on a set of genes, and these genes will result in the production of proteins associated with cell division and cell differentiation. The next receptor we're going to look at are called G protein receptors. I am going to save that for part 3 of our lecture. I want you to be able to look at each of these receptor pathways individually, so I'd like to keep them in individual lectures. So this is the end of part 2, and we will pick up with G protein coupled receptors in part 3.